Good morning. It's January 15th, and this is your daily brief in science. Here's everything you need to know. The Ice Memory Foundation has created a global glacier-ice archive at Concordia Station in Antarctica to preserve Earth's atmospheric history as glaciers melt. 2 initial ice cores from Mont Blanc and Grand Combin have been placed in the vault, with about 1.7 tons of ice shipped from Trieste by icebreaker over roughly 50 days. Launched in 2015 by French, Italian, and Swiss research institutions, the project aims to consolidate ice samples worldwide for future study when technology advances. A core objective is to keep the archive neutral and accessible for future scientific merit, governed by international law and open to proper beneficiaries amid diplomatic and legal considerations. The cores hold potential benefits across biology, chemistry, and geology, including preserved DNA, atmospheric gas records, and dust or mineral particles, underscoring their broad research value. The project operates on the premise that preserving these samples will enable increasingly detailed insights as technology evolves. Plans call for expanding to 20 glaciers within 20 years with global participation from nations, institutions, and funders to support further drilling and archive growth. Experts warn that glacier loss may peak around 2040, making rapid preservation essential to maintain environmental archives for studying historical pollution, atmospheric composition, and climate signals. Overall, the aim is to safeguard humanity's climate memory and provide a resilient resource for understanding past and future climate changes amid rapid warming. The initiative highlights the urgency of documenting climate records before they vanish, emphasizing international collaboration in climate research. Experts estimate that these cores could endure for decades or even centuries without melting, highlighting their long-term value. The effort seeks to protect climate heritage against glacier loss and rising temperatures, noting that 2025 was among the hottest years on record. A woolly rhinoceros genome has been recovered from tissue found in the stomach of an Ice Age wolf buried in Siberian permafrost, enabling a genomic look at the species. Researchers conclude the extinction was likely rapid and driven by climate warming rather than human hunting. This new evidence contrasts with earlier genomes from about 18,400 years ago that suggested a healthy population, indicating a swift decline for woolly rhinos in northeastern Siberia during the late Ice Age. The study, led by J. Camillo Chacon-Duc and colleagues and published in Genome Biology and Evolution, notes population-level data are scarce due to few young rhino specimens. The sample is among the youngest woolly rhino specimens found and presents challenges from degraded ancient DNA and predator DNA contamination. Recovering genomes from unusual contexts, such as a predator's stomach, can shed light on mating patterns, population dynamics, and extinction drivers relevant to conservation. Results showed a surprisingly stable genetic pattern over tens of thousands of years, with no clear signs of inbreeding despite small population sizes. The findings imply climate changes at the end of the last ice age likely triggered the extinction, offering historical context for current conservation concerns. Future work will focus on further analyzing the recovered data and applying the techniques to other difficult specimens, acknowledging the scarcity of young samples. The study demonstrates the feasibility of reconstructing an entire genome from DNA preserved in an animal's stomach, opening avenues for future genomic work from indirect sources. Leading researchers include Camillo Chacon-Duc and Solveig Gudjonsdottir, highlighting the importance of recovering genomes from extinct species near their extinction events. An effective population around 1,600 would have been sufficient to avoid major genetic problems, though small populations remain vulnerable to environmental change, disasters, and disease. A sweeping new global assessment finds subsidence is occurring in nearly all deltas studied, with some areas sinking faster than sea level rise and subsidence already exceeding local sea level rise in 18 of 40 deltas, raising near-term flood risk for more than 236 million people. The Mekong, Nile, Chao Phraya, Ganges-Brahmaputra, Mississippi, and Yellow River systems are among the most affected. Researchers mapped surface elevation change at about 75 square meters per pixel using high-resolution satellite radar, delivering the first delta-wide, high-resolution view of elevation loss across 40 deltas. groundwater storage loss, reduced sediment flux, and urban expansion emerge as key drivers, with a random forest model showing groundwater depletion as the dominant predictor for subsidence. The study emphasizes that subsidence is already occurring and driven by human choices, pointing to policy and management changes such as sustainable groundwater use and sediment management as part of the solution. Population growth and rising water demand amplify subsidence through increased groundwater pumping, while dams and river control measures curb sediment delivery, reducing natural compensation. The main causes identified are groundwater withdrawal, diminished sediment supply, and rapid urbanization. Mitigation suggestions include reducing groundwater extraction, recharging aquifers with floodwater or treated wastewater, using controlled flooding and sediment diversions, and restricting heavy infrastructure in subsidence-prone zones, alongside flood protection and climate adaptation. Policy relevance is high. Delta-wide subsidence and groundwater management must be integrated into coastal adaptation strategies and risk assessments. Urban growth and reduced sediment delivery contribute to sinking, with groundwater pumping singled out as the most influential anthropogenic factor across deltas studied. Subsidence, driven largely by groundwater extraction, is a major driver of land loss, coastal flooding, and saltwater intrusion at deltas. Groundwater depletion emerges as the strongest overall predictor of sinking, though drivers vary by region. A flexible OLED that stays bright when stretched sounds like science fiction, but a multinational team has demonstrated a device that preserves luminescence up to 1.6 times its original size. This performance comes from a combination of a flexible XIPH layer with MXene-based transparent electrodes designed to resist deformation during bending and stretching. The study, led by Seoul National University and Drexel University, focuses on integrating an XIPlex-assisted phosphorescent layer with stretchable, transparent MXene electrodes to tackle durability limits in flexible OLEDs. Full details are published in a Nature paper linked in the report. Researchers assessed electroluminescence characteristics, transient responses, and recombination dynamics as the device underwent strain. Extended data figures examine the photophysics, morphology, and energy transfer of stretchable XIPH-containing layers under mechanical strain, including high-tensile strains. The study compares XIPlex-forming co-hosts against XIPlex-free systems to see how deformation impacts electroluminescent efficiency. Funding comes from NSF and the National Research Foundation of Korea, with authors from Drexel, Seoul National University, Kyushu University, University of Tennessee, Yonsei University, and others. The XIPH layer is an XIPlex-assisted phosphorescent material that converts a large majority of excitons into light, surpassing 57% efficiency versus 12 to 22% for conventional polymer layers. In effect, the XIPH layer boosts exciton usage to over 57% of generated excitons, increasing light output relative to traditional OLEDs. Put simply, XIPH enhances both exciton formation and emission, achieving over 57% exciton-to-photon conversion. The XIPH layer is embedded in a thermoplastic polyurethane elastomer to improve mechanical flexibility and longevity. A new study points to regulatory factors in early embryogenesis that could fine-tune embryo development and shape next-generation assisted reproductive technologies. Researchers combined inhibitor screening with RNA sequencing data to map a regulatory network guiding the first steps of development, identifying factors not previously recognized. A Kanazawa Medical University team led by Associate Professor Hirofumi Nishizono and graduate student Masaki Kato identified 11 novel factors essential for mouse zygote development, using a multifaceted approach that included one-cell embryo cryopreservation, inhibitor screening, RNA sequencing, and CRISPR-Cas 9 gene editing. The findings, published in Frontiers in Cell and Developmental Biology on October 17, 2025, emphasize the study's comprehensive methodology and the discovery of critical factors underlying early zygote development. Among the identified factors, two molecules, CXCR2 and CTSD, were chosen for functional validation. CRISPR-Cas 9 knockout of either gene arrests development at the pre-implantation stage, underscoring their essential roles in early embryogenesis. The study relied on a large cohort of genetically uniform mouse zygotes cryopreserved as single cells, enabling high-throughput inhibitor screening and integration with transcriptomic data to pinpoint regulatory factors. The research is documented with a DOI and published in 2025 in Frontiers in Cell and Developmental Biology. Infertility affects about 1 in 6 people worldwide, highlighting the importance of understanding molecular regulators of fertilization, early development, and implantation to improve ART outcomes. Future work will address limitations such as inhibitor concentration and cryopreservation-induced stress, and will expand screening with broader chemical libraries to pursue molecular-level control of embryo development for infertility treatment and developmental biology research. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.